Thank you, Eileen. Okay, so earlier this morning, I gave you 3 asks, right? So the first was to go visit Kristen at the advocacy table about contacting your legislators. I was so happy to see that she was really, really busy before lunch, and there was a lot of people talking to her. The second was to join or update your surveys on the patient registry. And so thank you again, Eileen, for that great presentation. I know many of you will be at her table later. And the third was I asked you to text PKU to 41444. And we have already surpassed our goal. I know, I need to set my goals higher. But I have a new goal for you. How many of you were in Salt Lake City for a conference there? Do you remember when we did this in Salt Lake City? In Salt Lake City, we actually raised $22,000. So our new goal is to surpass Salt Lake City. And the $16,000 we've been able to raise so far, that has come from 80 different donors. And so let's get to 100, and we'll see where that brings us. The other thing that I think is really cool is you might see as some of these scroll, we have one donor who gave us $83. And I saw him, and I'm like, why $83? And he said, well, with that text to give, you can actually choose a monthly contribution. And $83 times 12 gets to $1,000 a year. And then you also become a member of the Ashburn-Furling Society, who actually Geyer is our honorary chair. And so if you were inspired by Geyer's story today and about Ashburn-Furling and have the means, perhaps not all at once, but you can spread it over 12 months, I challenge you to give $83 a month for a year in celebration of 10 years of progress. This is a great segue into our closing panel for the day, because you're going to be hearing from our funded researchers. So these are people that you have helped us fund for at least the last 2 years. They're going to give each a brief overview of their current research. And then tomorrow, all of them will be in different breakout sessions if you want to hear more about their science and the research and how our dollars are supporting that work. So at this point, I would like to invite Mike Franklin to come up, who will be facilitating the panel. He's the chair of our scientific advisory board. And then I also invite up Dr. Katherine Durand, Dr. Roberto Gramignali, Dr. Eileen Jaffe, Dr. Robert Nichols, Dr. Eric Copps, Dr. Carrie Harding, and Daylene Richards, Dr. Izu Dung. So please come up.
While everyone's getting up here and getting settled in their seats, I just want to make a couple comments. I've been part of the board since the beginning and was at the first conference. And when we started down the research path, our first year, 10 years ago, we had 6 applications that came in. And of those 6, we ended up funding about $130,000 worth of research, which coming from a local organization was a huge monumental event where we were lucky if we were able to raise $5,000 or $10,000. This last year, we had in the last couple years, almost 20 applicants for research proposals coming in, and last year funded over $500,000 of research just on this alone, let alone what we're doing with the registry and what we've been able to do with the home feed monitor and a couple of other selective activities. So when you think back on that, we've had over 150 applications that have come in to fund piloted research, and of that, 40 plus have been funded to generate new opportunities, new ideas, and new leaders in the field. So with that, I will start off with our first presenter. Now, just to warn you guys, we got about 10 minutes each, and we're going to try and give each of you a couple minutes for Q&A at the end. So I may give you the little speed up sign as we're going along. So the first one is Dr. Yuzing Dong, from Ohio State, and he's going to be talking about gene editing for the treatment of PKU.
Thank you so much. Good afternoon, everyone. It's really my great pleasure and honor to be invited for this wonderful conference. I really enjoyed it. And also, thank you so much for supporting our research. I don't have much talking about treating PKU yet, but I would like to give you an update about our research in the lab, and hopefully in the near future, we can apply that for the treatment of PKU. First, I would like to give you a brief overview about the research in my lab. So we are interested in the development of technology platforms which can be applied to diverse diseases. So for example, the first technology, it is lipid or lipid-derived nanoparticles. We can synthesize different lipid derivatives, and we can formulate them with different formulation components and deliver diverse therapeutic cargoes, such as messenger RNA or Crispr for gene editing. So the second platform technology we are working on is to engineer the Crispr system in order to increase the gene editing efficiency and reduce off-target effects. And the third platform we are working on is antibody or ligand drug conjugates, which is to target very specific cell populations and deliver therapeutic agents. We spend a lot of time working on messenger RNA-based therapeutics. Messenger RNA has become a promising class of drugs in the past decade. Messenger RNA are essential components in our human body, and it is now widely used in the clinical research and also clinical trials for protein replacement therapy, for treating infectious diseases, and also for treating cancers. But the delivery of messenger RNA is very challenging. You can see, even encapsulated in different types of biomaterials or nanoparticles, there are many barriers need to overcome, such as extracellular matrix here, and also even internalized in the desired cell populations, messenger RNA need to be released from the nanoparticles and produce the functional proteins. So to achieve all this, we need to develop new type of biomaterials and nanoparticles. So in my lab, in the past several years, we developed a nanoparticle system called OTT3. It's an optimized lipid-derived nanoparticles. We were able to utilize this material to deliver messenger RNA encoding factor IX, and for the treatment of model disease hemophilia. From this figure, we can see TT3 is able to produce factor IX in the mouse serum and also produce a functional factor IX. In normal humans, the factor IX activity is between 500 to 1500, and this OTT3 is able to recover the activity to 800. So that means this nanoparticle is able to fully recover the factor IX activity to the normal physiological range. And next, we utilized this nanoparticle for another disease model called hepatitis B. It is a worldwide pandemic. So we injected these nanoparticles with CRISPR-Cas9 gene editing system, and we were able to significantly reduce all the HBV biomarkers in this HBV mouse model. So right now, we are moving the project forward. We hope to finish the preclinical studies and move to clinical trials. And we hope to integrate our specialties in different research areas and develop more effective therapeutic medicine for patients in the clinic, including the PKU patients. With that, I would like to thank my lab members. This is a group of talented postdocs, grad students, undergraduate students, and high school students. Thanks our collaborators for their huge support, and also thanks for the funding agency, including National PKU Alliance. Thank you very much for your attention. I would like to take any questions. Thank you.
I don't think I see any there. Thank you. Okay, thank you very much. So all of the investigators will be out, like we had said before, afterwards. So if you guys do have questions or you're seeing any of the other information, please feel free to find those guys afterwards and ask those questions live. Our second presenter is Katherine Durand. And Katherine, I want to give just a little tidbit on. Katherine's one of the reasons why we have done some of what we're doing here. Katherine applied for a research grant long ago and actually got turned down. And as a result, part of our scientific advisory board actually gave her feedback. And as a result of that, she has now gone through and gotten her doctorate, been able to do a ton of great work, and is going to actually speak to some of that she's doing today, which is focused on probiotics and utilization in PKU.
I got turned down. I wasn't at a university yet.
Yeah.
All right, so several of you have been at multiple conferences. You're aware I'm trying to make a probiotic for treating PKU with an enzyme replacement therapy. I'm currently at the University of North Texas Health Science Center in Fort Worth in the lab of Michael Allen, and we are working with Trayer Biotherapeutics to try and get this into clinical trials now. So what have we done since we got funded back in, I think it was 2012? In 2017, we published our mouse results in PLOS One, which is an open access journal. That means anybody here can go read it if you want to. It is very science heavy, I'm sorry. But this sums up what we saw with the mice. So you'll see the dots on the left are each individual mouse, and it's got a line connecting where it landed after 1 week of treatment on the right. No, it does not bring it all the way down to normal levels, but every single mouse, with the exception of one that had a little bit of a pigging out issue, managed to drop their Phe levels. I was not very happy with him for that. So when we looked at the mouse version, and it went 2, okay. When we look at it in a Petri dish, one of the things we're looking at is how well does the enzyme work? And in this particular slide, the line that is going steadily up is the version with the enzyme that we engineered into the bacteria. The dots that are kind of in a straight flat line, that's with no enzyme added. So you can see when we add the enzyme, we got a lot of activity. We could measure what it was doing. But this is the mouse version. In order to do our work quickly and see if this was worth pursuing, we took some shortcuts that were not going to be okay for human use. So for the last 2 years, we've been trying to make the human-safe version. And I'm happy to say we finally have it. And we have looked at how the enzyme works again in a Petri dish. And you'll notice that the line looks very similar. We have a nice straight line. For those of you who understand the statistics, I've got the linear regression and the R-squared value up. We've got very good numbers. And if we go back, you see at 30 minutes, we had about 30 to 35 micromolar of tranexamic acid produced as a result of the enzyme functioning. If we go back at 30 minutes in the mouse version, we also had between 30 and 35 micromolar of product being produced. So its behavior in the Petri dish is very similar. We're trying to get the final batches made so that we can actually retest it in the mouse, because the Orphan Drug Association especially wants to see the final version and its safety profile before we go to people. So right now, despite the fact that using a lactobacillus slowed us down in many respects, we're very happy to be using it because of certain safety profiles we're noticing with other drugs using things like E. coli. We're not going to have those problems. We're awaiting our final preclinical batch, and we are at the point of requesting an FDA meeting so that we can start clinical trials hopefully by early 2019. I do have to disclose that yes, we've patented this so that we could move forward, somebody would pick it up and actually use it, and thankfully Trayer Biotherapeutics has done that. Mark Tui is here actually from Trayer to help answer some questions about how we are working on all of this. This button doesn't work very well. There we go. Special thanks to everybody in both of the laboratories I've worked in for helping us with the crazy things, the crazy hours, the times that I've been there. And, you know, I get there at noon and I'm leaving at 5 in the morning. These are the people that make that possible because they do the things during the more normal hours. I want to thank the National PKU Alliance and the NSPKU for their funding. Without it, we would have been stuck a long time ago. The ESPKU and the Canadian PKU groups have also been very supportive of our work. I will take questions. And one last thing, if you remember 2 years ago, we gave you this children's book title and said we are accepting samples because we are trying to understand the PKU gut. We are still trying to understand the PKU gut, and we made the sample collection process worlds easier. Technology has advanced, it's easier for everybody. And we did notice, thank you for all of you who gave us samples last time, there is a difference in the PKU gut. We're trying to figure out why. We need to confirm it, and then one of our goals is to hopefully see if we can fix it. Right now we are targeting PKU patients of all ages. We got permission for children this time, so anybody ages 0 through 65, if you have PKU, come find our booth. We would like to get samples so that we can see what's going on and maybe solve some of these tummy issues. That is it.
Hi, I was interested in what the results showed after 30 minutes.
Okay, so what are they showing?
Yes, you had your ascent of your enzyme activity, but that was only for 30 minutes. What happens after that?
Oh, so at 30, it's just kind of a cutoff point because what we've done is we've harvested the cells and we've broken them open so that we can see the enzyme and what it's doing. We use a plate reader. It has a max out rate, and usually not too long after 30 minutes, it hits the maximum read. If we diluted it and we kept looking, it would keep going up. It keeps working.
So, quick question: when you're looking at over here, when you're looking at your increase in product or enzyme activity, is that with a single dose? And then you mentioned the mouse that's a pig out, so I assume, was that like one feeding or is that repeated feedings?
So,
how often was it administered?
All right, so when you're looking at the linear graphs, that's in a dish, that's a culture, that's not a dose, that's just looking at the bacteria itself. When we are looking at, and I'm trying to get the back button to work and I'm not having any luck, so I'm sorry. But with the mice, what we were doing is we were mixing it in with their food so that they were constantly eating some of the bacteria as they were eating their food. It was always going in together. That was the easiest way to do it. How many people here have tried to get small children to take pills and not enjoyed that fight? Imagine trying to get mice to take something. So the easiest solution was just to mix it in with their food, and that way they were getting their dose constantly and consistently.
First doctor. I saw the results with the hemophiliacs. Do they use gene editing to treat the hemophiliacs now?
Yeah, right now we are working on that. For that treatment, it's a messenger RNA encoding the factor 9.
So,
And right now we are using the gene editing for that.
so you are treating patients right now with
Not
it?
yet, not yet. That's
No, not yet.
for mice, for mouse models.
Oh, okay. Thank you.
I had one more question for you. You said the PKU gut's different. How is the gut microbiome different from
So
normal
we had a
people?
really small sample size, and for those who aren't in science, the more samples you get, the more safely you can say what we found is not an accident. We only found one thing that was statistically significant, saying this is not an accident. We notice it's a microbe that's associated with gut inflammation, or when you don't have enough of this microbe, you see inflammation. So the fact that we see and hear from so many of the PKU patients that they have cramping, that they have abdominal pain, that they have digestive distress, kind of fits with that. So we're trying to get enough samples to make sure that we're seeing what we think we're seeing. Then we're going to progress to, okay, what do we know about this bug? How do we get it back?
I'm just interested in knowing if you have seen any connection between autism and PKU in terms of the gut wall and functioning of the gut. I know it's an area of research that, and I can't help noticing as well, a lot of families who have siblings or they have relatives who are on the spectrum. So I'm just wondering if it's an area that you've looked at or you potentially might look at.
We have not looked at that yet. We've started looking at specifically the one microbe that we noticed a significant difference on. It tends to be linked with inflammation. But while I do some of the work in the gut microbiome, that's not my specialty area. That would be, Viviana, where are you right now? Ask her.
Okay, so with that one, I think we'll thank you, Katherine. Our next speaker is going to be Dr. Robert Gramignale, who is coming from the Carroll Institute in Sweden and is going to be talking about cellular therapies in PKU. Another side note on Roberto, he was actually our first fellow that we started this program with. So once again, another huge success story of someone that we funded almost 10 years ago and now is back here speaking, not for the first time, but for multiple times about his research and all the great things he's doing.
Thanks. Thank you all for coming, and as previously said, exactly for supporting our research. So this is, you guys might remember me for the last time when I came and show you what we are doing. We are liver people. So for the past 25 years, we have been working in correcting liver disease, transplanting cells. So we use intact cells to inject to the patient. As I showed you last time as well, you might remember, this is a minor invasion because we inject the cell through the umbilical vein, and through that we reach the liver. In the liver, we release the cells, normal cells, not genetically treated, nothing. And we use this cell to correct inborn error like PKU, but also for rescue people with acute liver failure or for other disease. As you can see from this tiny schematic, we treated different kinds of disease, not only PKU. So far, in the past 25 years, we have been treating multiple different kinds of inborn error, including PKU, but mostly urea cycle defect and so on. So what we have done, and what I showed you also 2 years ago, was our results on the animal model. This job was done in collaboration with Kristen Zvorak, who's probably also in the audience. So we inject cells into the mouse. So this is a mouse with PKU. The phenylalanine level on the left is what we measure in the blood, in the right, what we measure in the brain. When we inject the human liver cell, we get a quite decent level of correction. But we don't have enough liver cell to correct all the patients we would love to. So we were forced to look for desperately looking for more cell sources. And what we found interesting was the placenta. That's what I'm going to talk about, because this is what's going to the clinical trial quite soon. Because when we inject human placenta in these mice, we got even a better level of correction. Not completely normal, but much better level of correction. So I'm talking about placenta. Placenta, I think everybody of you know that this is disposable tissue. This is something everybody throws away after they deliver a baby. So normal baby usually they come out together with the placenta is usually discarded. What we found out 10 years ago that we rescue some cells, in particular the cell from this size, the internal part of the placenta in contact with the baby, was originated by the baby at the very early. This cell and this cell only are capable to perform a lot of function, including maturing to liver cells, so expressing PAH, correcting PKU. And these are the cells we're collecting. You see the very dark pink disappear after our isolation, because those are the cells we collect and we use in animal and in the future for therapy. Of course, to perform this isolation, we cannot do it in the lab because we need to transplant to the patient, so we need to do it in a GMP. So you might figure it out a little bit how complicated it is. Not only quite unpleasant and not very fancy dress up, you know. I'm coming from Italy. I tried to convince Versace to give us a better one, but I didn't want to, so we wear this very uncomfortable white bunny suit, very hot, and we have to deal with a lot of regulatory proof. We have to work in GMP rooms, which are very expensive and very delicate. And of course, we need to use only biosafety material. Why we like this cell? This cell and this cell only are very, very interesting. I've been working with liver cells for most of my life and found this one very, very peculiar because, first of all, they're totally safe. They've already been transplanted in human. And none one ever report any side effect, inflammation, or even tumor, because that's the most important part. We don't want to cause a disease. We find out that this cell and this cell only are the only one capable to get a complete maturation into liver, so they can express liver gene, the one that most of our patients are missing. They secrete a lot of fat, growth factor, and so they support the other cell around. They are also anti-inflammatory. That's why it's been used for almost a century for skin problems and so on. But interesting, they express a lot of immunomodulatory properties. What does it mean? What is immunomodulatory? I didn't know much about it even 10 years ago, and I tried to show you very briefly and very simply. So every cell in our human body has to show a kind of identity card, pretty much the same as I have to show when I enter in this country. You know? You show your ID card, and then that in our cell is called HLA, and immediately the police is coming, exactly for us. No? In your body, you inject the cell, and then police is coming. If your ID card is wrong, more and more police is coming, and that's not good for your cell. Definitely is not. My cell, the cell collected from placenta, instead, they're peculiar because they express an HLA called HLA-G that is actually a shield. It's protecting this cell from the circulating cell, for the immune system, who's supposed to recognize the cell foreigner and reject them. They recognize it, but they leave it because they find it completely normal. That's why we were able to inject human cell in the mouse with no problem. So our cells essentially are peculiar cells with a lot of property and with this shield. So that's why, as all the young people in the audience might recognize, I think our cells are pretty much like this guy, with one very important difference. These cells don't have one shield. They have two, they have three, they have four different kinds of shield to protect this cell from immunorecognition, to avoid this cell to perform the function. And these are the molecules involved that we'll be more than happy to show you tomorrow if you want to come to the breakout session to go a little bit more further in detail to show you why they're important and why we identified that. So I'm waiting for you guys tomorrow, or if you want to stop me in the lobby, or every time I'm sipping coffee, I will drink a lot of coffee because of jet lag. So stop me by, ask me whatever you want. You pay for this research, so you need to know everything. So I'll see you tomorrow. And let me just finish say that this work has been started when we were in America, almost 10 years ago, as we said. And then it's continued when I moved together with my mentor in the other side of the ocean, in the cold Sweden, where, cold and dark. So we're still working there, and thanks to the whole collaboration we established, we are able to move this to the clinic. So right now, we have pretty much all the regulatory approval. So we're going to start probably a clinical trial in the next 6 months, because we are going to create the first bank, because we can bank this cell, we can save it, cryopreserve, to distribute to everybody in the world, and we're going to create it now. Thank you for your support and for your attention.
What did you say about when you'll be able to inject these in a human subject?
Okay, so right now, as I mentioned, we have to isolate in a very delicate way with this fancy room. And what we've been working, we have been working in a sort of way to submit all the documents to get approval by the European FDA, essentially, and the Swedish FDA. So we got the approval by the ethical board, and I'm waiting for the final approval for the Swedish FDA that's supposed to come in September. In September, I'm going to start paying this room and get this cell banked. Once I have at least a couple of billion or 3 billion of this cell, that's required at least 2 or 3 months, I might be able to transplant the first 10 patients. That's what I hope to. So likely, I guess, at the very early 2019. Unfortunately, not only due on our work, because we need to recruit patients. I'm going to talk a little bit more tomorrow as well. So there is several our team is quite big. We need a pathologist, we need the surgeon. So we need to identify the patient, and we need, more important, to get completely covered by the, sorry to say, but the expenses too, because I need to pay those people to work as well. So likely 6 months, likely. I hope so. Yes,
In order to ensure the long-term efficacy of this kind of treatment, I assume it's got to either have a sufficiently large number of cells transplanted, or you've got to be able to have some sort of selective advantage of those cells so that they persist post-transplantation.
Oh, wow. That's
Is
a very good
there
question.
some form of selective advantage that you could see being added to these cells in order to ensure that they persist and proliferate?
Excellent. You use a perfect word for that. So I try to make it very brief, but this cell, as you said, they have a selected advantage because they are totally normal. So when we inject in their environment, so the liver, where they're missing the function, they find the perfect way they survive and they express their function. We know that to correct many diseases, PKU included, we don't need to transplant the whole liver. We just need 5–10% of the liver mass. That's what we've done for other diseases. So we inject a certain amount of cell, and your question was also related to the amount of cell. And right now, we do not have the answer because we've been using in the past liver cell. But these placenta, they are also a little bit more, they're stronger. They can proliferate. So we exactly don't know how much we need. We might need a little bit less than we were supposed to. But let's stay with the worst scenario. Probably we need 1 billion of cells, 2 billion of cells, a little bit more, depends on the patient, to inject. Once they're there, they are able to stay in the liver, and we prove it. We are publishing how we transplant the cell to be certain they go there and stay there. They not only keep to mature, because these are the only stem cells you can find in literature right now that have been shown to express such a level of maturation into liver, they can correct the disease. The animal we treated, they will die otherwise. They cannot survive. So the best aim is, okay, they survive. Let's see how they can do that. And we did it quite efficiently. For safety, I'm quite confident about that. That's what keeps me saying quite awake at night, because I want to be certain to transplant only something that is very safe for the patient.
My understanding about cell transplantation is that children may be better candidates than adults because of the quantity of cell needed. Will children be considered in your trial?
Okay, I don't see. Oh, okay, I got you. Yes, they are considered absolutely, because in the past we transplant a lot of children. Pediatric patients are usually responding even better because they're a little bit more plastic and we need less cells. They are definitely considered. When we transplanted liver cell in Pittsburgh, we need to sign consent, so we were forced to select only 18 or older. In Sweden, it's still under debating, because the Sweden government actually allows us to use pediatric patients as well. So we need to use it's mostly related to the surgeon and to the pathologist, because we are working under hospital exemption. So we are actually, yes, think about to list a patient. Some of the disease we're treating, actually, they're fatal, they're lethal in the very early stage. So most of the time we hope to transplant patients immediately after burn. For PKU, every kind of age should be acceptable for us, absolutely, yes.
Yeah, I just noticed on this slide you had a list of other disorders, and one of them was clotting disorder. Is there any relation or association with other disorders in PKU, or has anybody looked into that?
Sorry, if I understood right, you mentioned the first slide when I mentioned all the disease we treated so far?
Correct. One of them was a clotting disorder, so I was just wondering if
the easiest correlation you can find with them is most of the situation is a monogenic disease. So it's one gene missing, like PKU, so it's one genetic defect we can correct. So most of the cases are different disease, not related to each other, except for this common fact. So it's urea cycle defect or bilirubin conjugation, completely different onset, but monogenic. And those are very effective for cell therapy. They show the best result in that. With transplanting also disease, they're not related to that, but mostly because we transplant cells capable to perform a function. That's it. Thank you.
Okay. Thank you, Roberto. Okay, our next researchers, I think, are going to maybe do a tag team here with Dr. Harding and his doctoral student, Daleen Richardson, to talk about CRISPR and gene correction in PKU.
Thanks, Mike. So I'm Dr. Kerry Harding from Oregon Health Science University in Portland, Oregon. So I'm my job as the principal investigator is to simply thank the NPKUA for funding us, because it's invaluable. This is the primary support for this very novel and early preclinical work in the PKU mouse. But I'm going to turn over the primary presentation to Dalen, who's the graduate student in my lab who's predominantly doing this work, and because she gives an award-winning presentation on this, much better than I would do. So she's going to give a very general presentation today and give most of her data on the results tomorrow. So Dalen, please.
Hey, y'all. Gosh, it feels good to say that. So I'm from Texas, and up in Oregon, I get a lot of strange looks saying y'all, but it's nice to be in company. So today, I'm going to talk to you about my graduate work, and I need to reiterate that all of this work that I'm going to show you today is funded by the National PKU Alliance. So it's gene therapy. Gene therapy is kind of self-explanatory, but let me explain it to you in a way that's easier to understand. So it's using genes, or genetic material, DNA, as a drug or a therapy. And what we do in the lab is we design a therapeutic DNA. And how we deliver that DNA to the cell is by use of a virus. Now, virus sounds scary, but has anybody in this room heard of adeno-associated virus? I wouldn't expect you to, maybe except for the gentleman over here. But it's because it doesn't cause any human disease. We call it non-pathogenic. Instead, we harness its ability that's evolved over thousands of years to do, which is deliver DNA to the cells of the body. So we package this therapeutic DNA into this virus, and we inject it into our preclinical model, which is our PKU model mouse, at which point the virus will then be able to deliver the DNA to the target cells, which for PKU is going to be the liver. Now, when it comes to gene therapy, there are a couple of different flavors. There's one approach called gene addition. And in this approach, we are adding to the system a functional copy of the phenylalanine hydroxylase gene. And in this system, the AAV virus delivers the PAH gene as free-floating circles in the cell. And the cell can use these free-floating circles of DNA to make a functional enzyme, resulting in a pretty robust therapeutic effect. But there's an issue with that, and that's because our liver is continually dividing and maintaining itself, and when the cell divides, these free-floating circles are lost. So this really good therapeutic efficacy that we can see from gene addition in the context of a mouse up to a year is inevitably temporary. And what does that mean for humans? We don't know. But when I came into this lab, I wanted to address this temporariness, if you will, and design something that would be more permanent. And to do that, what we decided to do is a gene editing approach. And for gene editing, you can think about it like this. We're delivering DNA to the cell that encodes for machinery. And this machinery can then go into the DNA and repair the mutation that causes PKU. This repair is within the genome, and it's there to stay, and it will divide. As it divides, it will deliver that, or it will maintain that corrected mutation throughout all of the future generations. So what I'm trying to say poorly is that it's permanent, and that the therapy in the cells in which this repair happens will not be lost. So I'm talking about gene editing, and at this point, I'd just like to introduce you to the technology that we are using to perform this gene editing, and that is the CRISPR-Cas9 system. Now, this is a revolutionary technology in genetics, and for those of you that haven't heard of it, I'm going to explain it to you. So think of it simply like a doctor performing surgery, and the doctor guides the scalpel and tells the scalpel exactly where to cut. Similarly, with this system, we provide the Cas9 enzyme a molecule. And this molecule directs the Cas9 enzyme to a very specific location in the genome right next to the mutation site, at which the Cas9 enzyme, or the scalpel, will then slice the DNA. This slice in the DNA allows for the cell to repair this particular location of the genome with the correct sequence of the DNA, resulting in a CRISPR-Cas9 mediated repair of the genome. So with that, I can tell you that we have some very promising results in the preclinical work that I'm going to be sharing with you tomorrow. And I'm sorry this is kind of a teaser, but I think it will motivate you to come to my talk tomorrow. And I can tell you this, that in our mouse model, who have an average of untreated PKU on normal standard mouse chow, so the equivalent of normal diet, they're around 40 plus milligrams per deciliter, so a very severe PKU. And in our CRISPR-treated animals, we have results with this particular approach of 10 and under. So it's a very promising approach, and we're very committed to advancing the dream for PKU, and we're very thankful to the National PKU Alliance. Thank you.
I'll be quiet if others want to speak up. So when you're looking at editing, and you're talking about just changing and switching out, I assume you're looking at individual mutations, not necessarily the whole gene sequence?
Correct. This is an isogenic model for this particular study. So this is all the same mutation, but we have come up with designs, and we are going after a more broadly applicable approach that would be applicable to all PKU genotypes in humans.
Would that broad approach be to just switch out the gene completely, or is that to isolate or sequence each individual mutation and then use the correct guide RNAs to get there?
Am I allowed to talk about this?
Talk about it. Go ahead.
Yes?
Yeah.
Sorry, I had to get permission.
That's okay.
Actually, our idea was actually to incorporate the coding DNA for the PAH enzyme in a targeted area of the genome.
Okay, so just inserting a good copy somewhere else rather than switch it out.
Correct.
Thank you. It's all been very exciting to hear what's going on.
Thank you.
Also the other talks, but I'm curious about this. What's next? Like how many more steps and studies, and what has to happen to this become reality? I have an 11-month-old PKU girl at home, so is she ever gonna? I think many of us have that question.
Well, okay, so that's actually a really good question, and not the first time I've gotten it. So in order to make this a reality for everybody in this room, there are some steps we have to take. One is addressing the fact that we have to make this approach broadly applicable to all PKU genotypes, because we want this therapy to be available for everybody, not just for a specific mutation. And the next thing that we need to do is to perform that study in our preclinical model. And then on from that, we have to make sure that our levels are in the normal range, because we're not going to design a gene therapy just to get us to a partial correction. We want to design a gene therapy that will be a full correction, and then scaling it up from a mouse to a human. So those are a lot of hurdles, but our lab is committed to doing that.
As you look to develop these cassettes, if you will, to go and find specific alleles. There are over 900 alleles for PKU and many combinations. Will you start with, say, the most common and work your way through that?
No.
And what does the regulatory path look like?
So that's a good question. You're right. There are 1,000 pathogenic variants of PKU, and to overcome that, we're actually looking at inserting the entire coding DNA of the phenylalanine hydroxylase enzyme into the genome. So you can think of it like a combination of repair and addition at the same time, as I think is what our next steps are going to be.
What's your timetable and expected budget?
Well, let's see. So I was able to perform this preclinical work, and I'm a fourth-year graduate student, and to design a new study and execute it and get it to where it needs to be, probably the next graduate student. And and then moving it into industry, of course, and into the clinic. So I'm sorry I can't give you like a distinct timeline, but I would say that this is a very exciting time for gene therapy, and I will say that it's kicking off and it's becoming a reality in our country. We just had the first approved gene therapy in the United States, and it's happening, and I think that this gene therapy is going to happen in our lifetime for sure.
Thank you. So for those of you, keep asking your questions. The one thing is I'm noting who's asking a lot of them, so you may get picked on later for maybe a scientific advisory committee opportunity.
Yes,
so,
absolutely.
Nothing like reading a bunch of a lot of applications. Our next presenter is Dr. Elaine Jaffe, and she's from Temple University, here to talk about pharmacological chaperones and the potential use and treatment of those in PKU.
Good afternoon. I'm going to start with 2 corrections. My name is Eileen Jaffe, and I am a member of the Fox Chase Cancer Center, which was acquired by Temple University about 5 years ago, and that may not be a permanent acquisition. Okay, what I'm going to talk to you about is some of the basic science research that was alluded to this morning. My title is Understanding Phenylalanine Hydroxylase Structure and Function and Dysfunction as it relates to PKU. And I have to figure out what button to push. Okay, this is a structure that you've probably all seen. This is the structure of phenylalanine hydroxylase. It was predicted 20 years ago by the pioneering work of Ray Stevens' lab. And we finally solved the full-length structure a couple of years ago. The first author of that study was Amelia Arturo, and she's here in the audience. Emily, can you stand up? I'm assuming you're here? Well, it's not standing up, but it'll have to do. So what I've done here is I've colored the 4 chains of this protein different colors. This is her crystal structure. If you go into the PDB, you can find it. It's called 5-DEN. I don't know why they name these things the way they do. And you can look at it and spin it around on the screen and have fun with it. So it's 4 sequence identical polypeptide chains. They're held together mostly by that 4-helix bundle in the center where you see the blue and the yellow and the red and the green coming together in a little circle in the middle. The assembly is mildly asymmetric. I'm not going to go through that detail. And this is the rat structure, which was solved 2 years ago. The newly determined human structure is essentially identical. It has somewhat more disorder. It's a little different in some interesting ways. And Emily has a poster on that, which will be a little bit later today. So what we now know about phenylalanine hydroxylase is it needs to fluctuate between 2 rather different structures, not just the one that I showed you on the previous slide. So in this illustration, I've colored 2 of the chains as white balls, and I've colored the other 2 chains, well, I colored what's called the regulatory domain. This protein has 3 domains. I've colored that magenta, and the rest of the protein I've colored in cyan. And what you see here in this known structure of the rat or the human protein is that there is a region, it's not so easy to see the purple, but there's a region of that regulatory domain that scoops down over the rest of the protein, which is where the catalytically active part is, where the protein takes phenylalanine and metabolizes it to tyrosine. And those circles show where that part of the regulatory domain, it's called the autoregulatory region, partially occludes the enzyme active site. This form of the protein is a low activity form for all of us, those of us who have healthy PAH and those of us who have dysfunctional PAH. There is this resting state low activity form of the protein. Now, what we proposed, was 2013, was that this protein had another structure. It had a structure that was activated, and here the big change that you see is that the purple parts now touch each other. They've moved. They've rotated about 90 degrees. And because the regulatory domain has rotated 90 degrees, now that active site is wide open. What you see is the gold ball is the non-heme iron that's at the active site of phenylalanine hydroxylase. So that's one way that the activated protein is different. The active site is wide open. The other way that the activated protein is different is it has a new protein-protein interface. And this new protein-protein interface, which I show with the black arrows, is the part of the protein where the allosteric phenylalanine binds. So there are two places on this enzyme where phenylalanine combined at the active site where it gets metabolized to tyrosine, and it combined at this allosteric site. And binding at this allosteric site would lock the protein in that activated conformation. It will stabilize the activated conformation. So it was a fabulous piece of work that came out shortly after we published the crystal structure of the low activity resting state form of the protein. It was a crystal structure, which is shown here just in the green circle, just of that portion of the activated, just of that portion of the regulatory domain that comes together. This portion happens to be called an ACT domain. We had proposed in 2013 that there would be an ACT domain dimer. It was unprecedented, and it was so cool when an entirely different research team, Wyatt Hughes' research team, published the crystal structure of that ACT domain dimer with phenylalanine bound just as we had predicted. How cool is that? Way cool. Okay, so that's our support for the activated form of the protein, but the problem is that that's really the only crystal structure that we have of that part of the protein. The rest of it is a model. It's probably close to reality, but the details are unknown, and those details are important. So the balance between the phenylalanine hydroxylase shapes, of course, responds to phenylalanine level. What I show here are tiny little pictures of my crystal structure, which can be differentiated by whether or not the purple parts are touching each other or not. In the pictures where the purple parts aren't touching each other, that's the resting state structure. That is the structure that predominates at low phi, which can be seen on my left side of this slide. But there's a little bit of that activated protein that's always around. It's in equilibrium. And as soon as the phenylalanine levels start to rise because you eat a protein meal, that phenylalanine then goes and binds to that activated form of the protein, stabilizes that activated form of the protein, and shifts the equilibrium so that on the right, what you see is predominantly the activated form of the protein with phenylalanine bound, and now very little of the low activity form of the protein, the resting state form of the protein. And of course, once it's activated, it starts to metabolize phenylalanine at the active site, the phenylalanine levels drop, and the equilibrium relaxes back to be predominantly at a low phi state. So this equilibrium, like any equilibrium, this balance will have a fulcrum. I imagine for my PAH, since I am not a PKU patient, that fulcrum is probably at around 50 micromolar phi. But for some of our patients, it's at 300, 600, 900, 1,500, And it's going to be different for the different genotypes. And because you see that this protein undergoes this massive conformational change, you can maybe start to understand why amino acid changes throughout the protein can potentially affect this equilibrium. So the tipping point of this balance is going to be related to an individual's basal Phe level. And again, because the shape change involves many parts of the proteins, this is one reason why there are approximately 1,000 disease-associated alleles. Okay, so the overarching goal, the long-term goal, the goal that we might not reach for another 12 or 15 or 20 years, because that's what basic science is. You heard about drug discovery this morning and the long timeline. But the overarching goal of our research is to discover or design new pharmacologic chaperones that will shift the tipping point of that equilibrium to lower or neurosafe Phe levels. And we want to shift that tipping point without removing the response to phenylalanine. So we don't want to permanently activate the protein. The envisaged drug might work independent from or synergistic with a drug like Kuvan. And a rational drug design approach requires that we know the crystal structure of the full-length activated form of the protein. And so that's the part of our work that the NPKUA is supporting. Thank you very much. And in the breakout session, I'm going to describe progress that we've made towards determining the structure of this activated protein. We've made this progress by designing PAH variants that will be covalently locked in the activated conformation. We've also designed variants that will shift the equilibrium so that the protein will be predominantly in the activated form of the protein. We've had some success with both of these methods. I'm happy to tell you about them. So not only will I talk about that work in the breakout session, but in the poster session later this afternoon, I have a poster up which talks just a little more about our view of PAH as functioning by an equilibrium between this resting state structure and this activated structure. So if you want to know more about it, we can talk about it this afternoon. Okay, so the acknowledgments. These are the primary people in my laboratory who have worked on this. First is Emily, whose picture is on top, just in case you didn't recognize her when she stood up. Michael Hansen, and at the very bottom, Elias Bourne, who is going to be starting medical school in the fall. We have a lot of we've used a lot of biophysical tools, and so we have a lot of collaborations, and many of our collaborations are mentioned here. I'm not going to go through them individually. And we've had a tremendous amount of support from my parent institution, the Fox Chase Cancer Center, before we became funded on this research. We have had some support from BioMarin Pharmaceuticals and by NPKUA, and I'm happy to say that we're about 2 years into a 5-year NIH grant to pursue these studies. So yay NIH, they're supporting our work. So I thank you for your attention. I'd be happy to take questions, but I have to tell you, I've had a hard time hearing the questions to other people, so you might want to leave them for the poster session or tomorrow's breakout session. Thank you. Oh, I have successfully gotten them deferred. Thanks.
Thank you. Okay, so our last combo speaker here is Dr. Nichols and Dr. Copes, who are from Pittsburgh and are going to be talking about our PKU pig model as well as how CRISPR technology is being applied in that forum.
Okay, what happened to it? Oops. There we go. My laboratory is interested in neurobehavioral and neurocognitive deficits, so the genetic basis for these, the pathophysiological mechanisms, and then how we might develop better therapies. And while my primary area of interest is a different genetic disorder, a number of my colleagues, Dr. Jerry Vockley and Steve Dobrowski, have worked on PKU for many years and may be well known to a number of you. And from that, this collaboration developed, and most of the work's been carried out, a lot of the work by Dr. Eric Copies, who's here, and he'll be presenting a poster, and we also have a longer presentation tomorrow. And so in PKU, if untreated, the phenylalanine levels are toxic to neurons. And it's thought that part of the deficits arise from defects in neurotransmitter synthesis due to this pathway of phenylalanine being converted to several other molecules and onto the neurotransmitters. There may be other mechanisms involved also, but key is that without very, very tight control, the phenylalanine toxicity can lead to neurocognitive or neurobehavioral deficits. And so we're interested in using animal models because it's very hard to perform research on human subjects looking at the brain and brain mechanisms. And so the reason we use animal models is to get a much more detailed biomedical understanding of the complexity of the brain. And it also allows testing of new therapeutic approaches which cannot be performed directly in human subjects without a preclinical model. This talk is about the pig as a model, and as many may know, mouse is a common mammalian model. One of the issues with mice is that the brain is very small, and if you look at the top left, you can see its cousin, the rat, which is brain is quite a bit bigger than the mouse brain, but it's still much, much smaller than the human brain on the bottom right. And what you can also note from the rat brain is that the cerebral cortex has a very smooth appearance. You can see this both on the whole brain and then the section to the right. Whereas the human brain is cerebellum has these large amount of cortical folds, and this is associated with greater functioning and capacities. And so if you look at two other possible animal models here, the rhesus macaque or monkey on the top right, or the pig, you can see that the pig brain is actually closer in size to human and has more cortical folding than even the rhesus macaque, which is a non-human primate. This suggests that the pig should be a very good neurobehavioral and neurocognitive model, and indeed the pig is widely regarded as being a highly intelligent animal. We've already had a really nice introduction to genome editing, the use of CRISPR-Cas9, and in my laboratory we typically like to make deletions to generate models of disease. If you make a deletion of a gene, you can be sure it's inactive, whereas some laboratories make single nucleotide mutations in the DNA, and then you're not sure if you have an inactive or partially active or even functional enzyme. So we use the Cas9 enzyme, which is actually derived from the immune system of bacteria. They actually have one based on this system. And it's basically a molecular scissors or scalpel, like we heard before, that allows the DNA to be cut. And the way we get specificity is from the RNA component, the Crispr, which acts basically like a zip code to target the Cas9 enzyme directly to the gene of interest. And so in doing this, in the case for a model for PKU in the pig, we were able to target a single exon, one of the 12 parts of the gene that for deletion. And so we made 2 cut sites either side of this single exon, which leads to a frame shift in the coding sequence and a non-functional protein. So this is a null mutation. And the way we as geneticists can recognize a mutation is on a gel at the bottom. We're looking at DNA, and the top band is a normal band. It's basically like that picture of the nice bridge at the top left. It's just fine. You can drive across it. It works good. But all those bands at the bottom that are smaller, those are the deleted DNA fragments that we've specifically detected by PCR, and they're equivalent to the mutation. The bridge on the right you can see is broken. So that's how we can recognize mutations. So to generate a pig model, you take the RNA components for the Crispr RNA and the Cas9 enzyme and inject the RNA encoding these into a single fertilized zygote, so one cell stage. These cells are then cultured in the laboratory for a week and then implanted back into a female, and she then hopefully becomes pregnant. We did 5 embryo transfers and had 2 successful pregnancies to generate offspring. This represents, actually for the PKU project, we had 1 successful pregnancy, and we obtained 2 animals shown here. We've named them just in the laboratory, 116-1 and 116-2, and from many detailed molecular analyses, we've developed the map at the bottom, and Dr. Copies will present much more about this in his poster session. The 116-2 is a heterozygote, so it has 1 mutant copy of the gene, the other is normal, and so she should be normal clinically. And the 116-1 is what we call a compound heterozygote. There's 2 different mutations that one just happened to be slightly larger in size. And as we can see here from the blood phenylalanine levels, 116-2 indeed had a normal blood phenylalanine level of 141 micromolar. It's completely normal for a pig, whereas 116-1, the PKU model, the blood phenylalanine at this first test was over 2,000 micromolar. And we've done several tests since, and it's even a little bit higher than that. And she also excretes urinary phenylalanine and metabolites, as might be expected. The PKU pig, 116-1, has hyperpigmentation. This is expected from the role of phenylalanine in the biosynthesis of melanin, the pigment. And at the bottom is a growth chart. The PKU pig is in blue, and she has some growth retardation compared to her normal sibling. And these are some recent pictures, just like a little photo album. Interestingly, we've just started we've just imported these animals from our collaborators' laboratory in Missouri to Pittsburgh and started working with them. We've made the observation that while pigs prefer to use a pressure activated water nipple, which is thought to be because they prefer this to bowls or buckets of water which get dirty. Again, pigs are smart, they like clean water supply. Our PKU pig has never figured out her water nipple and only drinks from the bucket. And so the next slide would show a quick video. This is Eric took this, and this is Eric setting up the bucket of water. This is 116-2, the heterozygote, and she takes a sip of water, pops her head in the bucket, drinks her water, a little bit like if you have a dog, like your dog. And then quickly, this is 116-1, the PKU. She dips her head right in the water, leaves it in there for some time. She kind of blows bubbles, has trouble drinking to the same rate. It takes her a lot longer to do so. So this could represent, we have to perform detailed analyses, but some type of learning disability and or neurological deficit. And so in conclusion, we've been able to generate a pig model of PKU using efficient gene editing in zygotes. This will allow study of the biomedical basis, neurobehavioral studies particularly, and potentially collaborations to look at therapeutic approaches. And right now we're breeding. It's a lot slower than the mouse. That's one big deficit and more expensive. And then the people who did the work, it's a whole team, both in Pittsburgh and collaborators in Missouri. And thank you to NPKUA for their support of this project from the beginning to these animals. Thank you.
Okay, I'd like to thank our panel for everything that they've walked through today. And just to note, so the breakout sessions tomorrow, there are 3 different ones starting in the morning, running to the mid-morning, and then in the afternoon. And most of these guys will all be speaking there with more detail. Along with, I've seen a lot of you guys trying to take pictures of the slides. We will ultimately have those available on our website, as well as these main presentations here are going to be videoed, so you'll be able to have access to those. It may take us a little while to get those up on the site after the conference, but that will be access for those of you that want to share with your family or your constituents at home. And once again, I want to thank all of you who are out there who have been giving, because these guys, a lot of this work would not be possible if you had not, and your constituents and your different affiliates donated to make this possible. So thank you again.